Hello, everyone. So today we are going to discuss about the unraveling proteomic networks that we have built using Bayesian learning. So the applications are mostly in cancer and autoimmune diseases. So yeah, okay. So this is the lab where I work, where I'm doing my PhD. So in Mohan's lab, we focus on improving the lives of patients by discovering novel disease biomarkers related to specific diseases, especially lupus nephritis, IBD, bladder cancer, so both autoimmune and cancer related studies. And there's also a lot of omic data analysis that's being done. So let's understand what biomarkers and omics mean. So before that, the quick recap of my journey that I discussed yesterday. So I did my master's in mathematics and statistics, bachelor's in mathematics and statistics, and then master's in statistics from India. And here I am doing my PhD in University of Houston, which in biomedical engineering, but my background is completely statistics and big data analytics. So I'm trying my best to learn the biology here. Okay. So understanding omics and biomarkers. So if you look at the image, we see three segments: proteomics, transcriptomics, and genomics, right? So these are the omics study that I was talking about. Proteomics is the study of proteins, transcriptomics is the study of RNA, and genomics is the study of DNA. So when we have a large scale data of these biological molecules, could be genomics, proteomics, and transcriptomics. So when we look at these data and analyze them, that's the omics study that we are talking about. Now, biomarkers are the biological molecules found in the bodily fluids, let's say urine, stool, blood. So that's what biomarkers are. We come across a lot of biomarkers in our daily life. For example, we come across blood glucose levels, which is a biomarker for diabetes, and then cholesterol, which is a biomarker for cardiovascular health. But this is a single biomarker. But when it comes to lupus or any other cancerous diseases, we cannot detect any of this with just one single biomarker, but we have multiple proteins or multiple biomarkers that we need to study. So if we are studying proteomics, that's proteins, like thousands and thousands of proteins would be our biomarkers now. Okay. So this is the kind of data that we work with. So now imagine us moving from one single biomarker to thousands of proteins. So how do we draw meaningful patterns from this complex data that we have? There are two things that we usually do: the general biostatistics and machine learning algorithms that we apply in that probably predicts clinical status. Let's say there are subjects belonging to healthy controls and lupus. So what biomarkers are significant enough in predicting samples with lupus? So why do we have to do all of this? Because majorly to avoid invasive biopsy testings and things like that. This is one of the non-invasive ways to check biomarkers, draw patterns from biomarkers. So in regular biostats, what we do is we build volcano plots to see upregulations and downregulated proteins, like how elevated are they in the study. And then we perform ROC curves to get the top 10 with top AUC, like that discriminates between the two groups of data that we have. And then most importantly, we have our statistical tests, which gives us p-values when we compare two different groups. So that's how we generally get the significant biomarkers. Apart from that, we also use a Bayesian network using Bayesian Lab to uncover certain patterns between the data. They could be not just the proteomic data, but the proteomic data with its clinical and demographic data combined. So when we have three different categories of data, how do we observe patterns and molecular subgroups and finding clusters out of the data? So that's where we use Bayesian Lab. So it is a very powerful tool that helps us that where we can use supervised or unsupervised Bayesian learning to get or to analyze the high dimensional data set to uncover hidden relationships, clusters, and dependencies. Majorly in our lab, we use unsupervised algorithms that are available to understand the patterns running inside the data. So we have maximum spanning tree, we have EQ algorithm, we have taboo search, and all the analysis that's done focuses on these two metrics: the node force, which influences the importance of the node, and the correlation coefficient, which gives which talks about the strength and the direction between two variables. So this is a first case study on lupus nephritis. So in this, the sample which is taken, the samples are healthy controls and lupus samples, but the main question is, can urine proteins, so the proteins that are found in urine, predict LN status? So in this study, we tried to identify molecular and pathological correlations with lupus nephritis by basically integrating all the proteomic, clinical, and pathology data that we have. So if you look at the image, we see different colored nodes. So there are proteins with purple color, and then green and yellow are the pathology data. Red is the response, and the blue are the demographic data. So that fall under the clinical information. So this is we performed Bayesian network analysis using the EQ algorithm, which is unsupervised. And this is the kind of network that we could see where, yeah, okay. So we can clearly see that the LN5 has a negative correlation with endocapillary hypercellularity. And then we can also observe a group of proteins that are clustered together towards the end. So this is all with respect to unsupervised learning. So there is no labels that are actually defined. So everything, we just trying to understand the relationship between all the data nodes that we have. So that's how this is a part of analysis which was used in the manuscript that I showed earlier, which gave us information of protein clusters that we could further study in knowing the pathways and the molecular subgroups it belongs to. The second case study is about IBD and CRC. So IBD and CRC are basically the inflammatory bowel disease, and then CRC is colorectal cancer. So this is a comparison between three groups of samples. We have healthy controls, we have IBD, and we have CRC. And then all the proteins are in yellow color, and then you see the nodes with blue are the demographic data: age, gender, and race. So and then again, I think this was a maximum spanning tree which was used to build a network, and clearly we can see that the node, the disease status node, has a greater importance because it's huge, and also the clustered proteins around the node. Uh, yeah. Yes. So these are the clustered proteins around this node, which has negative and positive correlations. So we can say that when a sample moves from healthy control to cancer, in between the inflammatory stage is also there, but when the person moves from healthy to CRC, there is a negative correlation with the protein BIMP.7, which is basically down regulated. So you see that when you move from healthy controls to CRC, the BIP.7 decreases. So that's how we can interpret the positive and negative correlations, through which we can also understand the levels of these proteins in samples. If they are elevated, if they are downregulated, all of that could be studied. Um, so this is an individual comparison, not three samples together, but just IBD versus healthy controls and CRC versus healthy controls. And how do we check these results is mostly using the existing literature. We check if I mean, if we check if these proteins are already in the existing biological literature, and that's how it makes more sense for us to interpret it. So in this case, lactoferrinin is positively correlated with healthy controls and IBD, which means it's upregulated in samples with IBD and is downregulated in healthy controls. And there is also in this image the clinical data, like the location of CD is Crohn's disease. So the location, the type of behavior, and then there is gender and race. I mean, I see like there's not much significance, but there is correlation with certain proteins that we see. So this is the kind of analysis that's done, and that's about IBD and CRC, about can we predict IBD and CRC from stool. So yeah. This third case study is about urine proteome, which is basically the molecular taxonomy that I was talking about. So what is molecular taxonomy? Molecular taxonomy is basically to uncover new molecular subgroups of patients without using diagnosis labels, which is completely, again, unsupervised. So the traditional method is to have a clinical status of a subject, and then we try to, I mean, everything that we have done before earlier, we try to see which are the biomarkers that are more significant based on the clinical status of a subject. But the aim of molecular taxonomy is to find those molecular subgroups among patients without actually knowing their own clinical status. So the end goal is to probably, find pathways. I mean, if we are targeting any patients for treatment, instead of targeting them with their disease or their clinical status, we need to target them with the pathways that they have, which is more robust and uh, in targeting the samples. So yeah, in here, what we have done is we have 83, 85 patient groups that belong to different, different groups. We have DKD, we have healthy controls, we have urology controls, bladder cancer, and lupus nephritis. So these are the different groups of data samples that we have. So what we're trying to do is understand those pathways that are linked to these samples with the proteins data that we have without actually knowing the clinical status of these samples. So the analysis which was done was first we used walk-trap algorithm. We performed different clustering algorithms, but walk-trap worked best. We are still exploring more clustering algorithms in order to cluster the protein modules. So we used 7,000 urine proteome for this data, which was clustered using walk-trap algorithm, which created almost 863 protein modules, out of which, based on the module score, based on the number of proteins in the modules, we chose the top 15 robust modules. So those top 15 robust modules were used, and then the groups that we see above, there are 85 patient samples, which were again grouped using hierarchical clustering. So that was the groups that was done. And then the heat map that you see here is basically the groups in the x-axis and the modules in the y-axis. So this is the module and place patient clustering together that we can observe. Now, out of this, what we can see is the module 6. So if you see red, it means the protein levels are elevated. The blue means the protein levels are downregulated. So in module 6, we kind of observed significant shades of colors with respect to all the groups. So we wanted to focus on module 6 further. And if you see the grouping here as well, we can definitely see it's not clustered properly. There are mix and matches of different groups here, but it could be because there is some molecular significance with that group. There could be their pathways defined, which could be same for both, samples with both the disease status. So that's the main goal, that's the main aim. So targeting the pathways instead of disease status. So we then focused on module 6, and we extracted the proteins in module 6. I don't have the images here, but we did extract the proteins from module 6, and we did pathway analysis. So there is gene ontology, there are KEGG databases online, through which when we give a set of input proteins, it gives us the pathways that are enriched by these proteins. So that's the main goal that we are trying to see. So yeah, so that's the main goal, but then we are still exploring ways, because the scaling and all is also a problem. We need to like fix that too. So we are finding different methods to like accurate methods to proceed with clustering and scaling. But yeah, um, we also wanted to see how these modules are related to each other along with the clinical data and demographic data that we have. So that's where, again, Bayesian analysis. So in here, again, it was EQ algorithm, and then the group is all the samples belonging to the groups from healthy controls to bladder cancer that are grouped together. And then we have age, gender, ethnicity, and then we have cancer stage or CKD stages that were defined, and then these are the modules. Clearly, module 0 has a greater node force, so we wanted to check module 0 as well. And module 6 was what we identified, but module 6 is again correlated to module 14. So we wanted to like check further the proteins in module 14, how the pathways are enriched in this particular module. So that's the reason why we perform this. And we also, this is, the correlation here is quite less for gender and group, and for module 10 and gender as well. It's quite less, but we can see like a positive and a negative correlation. So I think in gender, male was 0 and female was 1. Sorry, female was 0 and male was 1. So as you move on from healthy controls to ALN, to lupus, to DCKD, and finally to bladder cancer, we can see that the gender, is also moving from female to male. So um, a lot of lupus patients in general in literature, 80% are women. But bladder cancer, again, depends. So that's an interesting insight. And again, gender is negatively correlated with module 10. We can see the sign, but the strength is quite less. But yeah, so this is an observation where we used Bayesian analysis, network analysis in the study. So yeah, so the biological insights that we get from Bayesian network is basically to uncover the hidden nonlinear dependencies in the complex proteomic data that we have. So we have been using supervised Bayesian algorithms. We can use, sorry, unsupervised algorithms. We can definitely use supervised algorithms too, but we just generally go on with the ML algorithms, execute them in Python, and then put it in the manuscripts. But Bayesian Lab, we have been using it mostly for unsupervised Bayesian algorithms that basically helps us generate some hypothesis about these disease mechanisms to have potential therapeutic targets on for these samples. But the future directions, what we are trying to do is one challenge is the high dimensional data scaling, because we separately analyzed proteomics and transcriptomics data until now, but the goal is to combine both the proteomics, the protein and the RNA information together. So that creates a whole problem of data scaling. So we need to figure out that, and after viewing all presentations yesterday and today, there is something that I want to rush back home and try applying, is the protein and pathway clustering. So we have just seen proteins, clinical variables, but now we go elsewhere to check the pathways. So I want everything to be on Bayesian Lab, where we can see all the data together, pathways and proteins and clinical data, everything together integrate it and yeah, study that. So that's what I'm planning to do now. Yeah, and then thank you.